That's what I thought. Okay. Thank you, everyone, for attending the Needham Healthcare Conference. It's my pleasure to introduce John Kuch, the CFO of Xencor.
Thank you very much for the introduction. Again, John Kuch, Senior Vice President and CFO of Xencor. Before I get started, I'd like to acknowledge our host, Needham. Appreciate the opportunity to present this afternoon. Here are forward-looking statements. I refer you to our Form 10-K, 8-K, and 10-Q for additional information with respect to any financial information presented today. So Xencor, what is Xencor? Xencor is an antibody company that uses our protein engineering capabilities to modify the protein structure to develop drugs with improved properties. Now we talk about what we typically do. Our focus is on the Fc part of the antibody. Here you see a standard antibody structure. And what we do is we make small changes in the Fc domain to create improved properties. Our focus is augmenting the native immune function, preserving stability, half-life, and production. And we talk about the changes we're making. Typically, it's 2 or 3 amino acid substitutions in various parts of the domain. Now we've got a number of Fc domains or technologies that we've developed off this, created a number of bispecific candidates for ourselves and our partners. We have a broad intellectual property estate, over 500 issued and pending patents that cover our technologies and our pipeline. The company's current focus is our expanding bispecific antibody pipeline technologies. There's currently 4 bispecific candidates in phase 1 development. XmAb 14045, 13676, 18087, and 2717 are all in phase 1 trials. In addition, we have 2 additional candidates have open INDs, which you expect to start clinical trials in the first half of this year. I would note that phase 1 study of 14045, our CD123 CD3, is currently on partial clinical hold with the FDA. We're currently working with the FDA to resolve that. 14045 is part of our Novartis collaboration. Novartis has ex-US commercial rights to this compound. We're co-developing that program with them and sharing costs 50-50. Finally, I point out, in addition to our own programs, we've got a number of partner programs in the bispecific space. Amgen has 2 programs in development, including AMG 424, which is a multiple myeloma CD38 CD3, which was developed under our collaboration with them. And there's a second preclinical program, AMG 509, which is developed in that collaboration that's going to be targeting prostate cancer. Now, we also recently announced an exciting license and development collaboration with Genentech around our novel IL-15 cytokine program. Under this program, we received $120 million up front. First IND under this program, XmAb 24306, will be submitted by the end of the year. This is a wide-ranging collaboration seeking to maximize potential with multiple combination strategies. An important part of this collaboration is we do retain the right to use the IL-15 cytokine on our own programs, subject to certain limitations. Finally, this broad-reaching platform, we have 12 clinical programs that are ongoing, either development by us or our partners. 2 most advanced ones are MOR208, which is being advanced by our partner MorphoSys. It's in phase 2 and phase 3 trials. They have breakthrough status for DLBCL, and they've guided they are going to submit a BLA for this with the FDA by the end of the year. And finally, Ultomiris, which is marketed by our partner Alexion, was approved for marketing in the U.S. in December of this year. This is the first compound approved for commercial sale that includes a Xtend or XmAb technology, so we're real excited about that. And down here you see a number of the leading partnerships that we currently have. Now, Xencor's focus is a little bit different than traditional antibody development. Traditional focus has been on the FB. So up here you see a suite of successful antibodies that developed. They all have different FBs, but they have the same Fc. Xencor focuses on engineering the Fc, or the constant part, and by focusing on different aspects of the Fc, we've created a suite of different properties and a suite of different candidates off of this. And one thing I want to point out is when we talk about small changes in each of these Fc domains resulting in different properties, let's touch base talk briefly about Soliris and Ultomiris. Ultomiris is the second generation of Soliris, which is marketed by Alexion for treatment with adult patients with PNH. Soliris dosing is IV every 2 weeks. When they generated a second or improved version of Soliris, they took most of the antibody Soliris and creating Ultomiris. One of the more important changes is they incorporated Xencor's FC extend domains in creating this antibody. The net result is Ultomiris is the same target population, but instead of IV every 2 weeks, it's IV dosing every 8 weeks. Small changes in the FC domain resulting significant changes in the property with drugs with improved properties. So that's the thesis of Xencor's protein engineering efforts. Now this slide shows the various flavors or different FC domains that we've worked on and the different properties. In all cases, we start out generally with the natural FC function, and here's the FC receptor for each of these various ones. And what we do is couple amino acid substitutions, and each of these results in improved properties. The green is our extend technology. The orange is our cytotoxic FC domains. The blue is the immune inhibitor. And finally, we use the FC domains to create a suite of bispecific candidates. Now, one point I want to point out of this slide is this also shows the continued evolution of our protein engineering efforts. The earliest efforts were 2003 to 2006 were around the extend and cytotoxic domain. We evolved and advanced into the immune inhibitor focus toward the end of 2000, and most recently, in 2011, 2012, we started working on the bispecifics. So this shows that the protein engineering efforts allow us to continually mine new opportunities to expand changes in the protein structures with improved properties. In addition, these technologies build off each other. So, for example, we use our extend domains in certain of our bispecific candidates. For example, our tumor microenvironment activators as well in our cytotoxic. We incorporate the extend FC domains for improved half-life. So again, this shows that the focus, the real value driver behind Xencor is our protein engineering efforts to constantly identify new ways to improve the properties. Now this slide shows the pipeline of candidates that we are advancing, and it's changing. As you can see up here, the top blue are candidates built off our Fc gamma 2b or our 2b technology, while purple is the growing focus, represents our bispecific candidates. There's currently 4 in the clinic, open INDs for 2 additional ones, and finally our XmAb 24306. We expect to assist Genentech with filing this IND by the end of the year. Now this slide represents the FC partnerships that we've entered over the years. One of the key benefits of our technology and our approach is a plug-and-play approach. It allows us to license access to our technologies or apply our technologies to partner programs with limited resources on our behalf. This allows us to generate upfront payments, milestones, and potential royalties from a number of collaborations. And as you can see, this includes the greens are extend, the orange are the cytotoxic domains, and in the purple represent the growing number of partnerships under our bispecific, including a most recently announced collaboration with Astellas that we announced last week around the bispecific. So this platform allows us to consistently build off these partnerships to generate ongoing payments, upfront payments and royalties. Even since the time we introduced our bispecific technology, we've generated 3 major collaborations which result in over $325 million of upfront payments and milestones. So it has a significant ability to bring in non-dilutive cash and allow us to use our resources on our own pipeline. Now, I'd like to touch briefly on our bispecific programs and technologies, since that is the key focus of the company moving forward. Now, what we've tried to do is, with our bispecific focus, is take our protein engineering capabilities and address the problems from the first generation bispecifics. There's always been the potential of bringing 2 antibodies together, either for improved potency or dual targeting mechanism. Now, some of the first efforts were Micromet, or Amgen with the BITE, and Blincyto. Very effective drugs, but a number of limitations. Specifically, very difficult to make. When they are made, they're not very stable, very short half-life, and the other problem is they're very potent drugs. Blincyto is a very potent drug, which means you have to limit the amount of drug that you can give patients. A combination of strong potency and short half-life requires the dosing to be on a continuous basis. So you're either using continuous infusion or in the way that Blincyto is dosed with the pump. So what Xencor tried to do is take our protein engineering efforts and address some of those problems. Now what we did was we took a full-length antibody and fused it with an Fc. The Fc part of the antibody is one that gives it its natural properties. It improves half-life, stability. Now on the other side, we put a light chain and two heavy chains, and we engineered the Fc part of that so that there was natural separation between the two antibodies. The net result is we have a stable bispecific format that allows us to have a bispecific with traditional antibody functions. So for example, it's able to be manufactured very similar to standard antibody production with CHO cell. Use third-party manufacturers for all our antibody bispecifics. We've now taken 6, 7 bispecifics, manufactured them under standard manufacture with third-party manufacturer without any problems. Stability, very stable, natural half-life, and addresses all the problems from the first generation. Now, by doing this, once we generated this format, basically we have a plug-and-play approach. You've got the two antibodies, the Fc. You're able to decorate each side and use the same format to generate multiple bispecific candidates. This allows us to generate a suite of candidates very quickly and efficiently. Last year, we submitted INDs for 3 bispecific candidates. The only way we're able to do that is by having a very efficient drug discovery capability that can generate this quickly and efficiently. We also use this format in our licensing collaborations. So when we have collaborations with Amgen, Novartis, and our recently announced Astellas collaboration, it will be the same type of format. It'll be a plug-and-play that we can apply with limited resources. So that allows us to generate a high number of candidates for ourselves and our partners without tying up a lot of resources. We don't have to redesign the antibody every time. It's basically using this format in a plug-and-play approach. Now, the first suite of bispecifics we generated are T-cell engagement CD3s. And what we have on each side of this bispecific you see is the CD3, which is a T-cell recruiter, which allows it to increase the potency of the other side. And we decorated the other side with the tumor antigen CD123 under our 14045, CD20 under 13676, and finally 18087 is SSTR2 CD3 bispecific. Again, these are all plug and play with CD3 on one side, tumor antigen on the other. Make them quickly. We've been able to manufacture them. They're stable, and they have an antibody-like half-life. And down here we see early animal models that show the potency of each of these CD3 bispecifics. For example, the 14045 showed profound sustained basophil depletion, CD20, profound sustained B-cell depletion in the animal model, and SSTR2, potent dose-dependent tumor reduction. So again, very quickly, plug and play, generate a suite of CD3 bispecifics. I'll touch briefly about our lead bispecific candidate, which is 14045, which is a CD123 CD3 bispecific. It's a full-length antibody to be dosed intermittently, as opposed to continuous infusion, which some of the other candidates are, other competitors are. Now, XmAb 14045 stimulates targeted T cells, presents CD123 targeted AML, and also leukemic stem cells. This antibody preserves antibody half-life, does not require continuous infusion. It's efficiently manufactured under standard antibody production. The phase 1 study is in relapsed refractory AML. It is on partial clinical hold. We're working with the FDA to resolve that. We presented encouraging data from this trial with 28% CRI at ASH in 2018. And here you see the preliminary data. 5 out of 18 evaluable patients presented showed either complete remission or complete remission with incomplete hematologic recovery at the two highest initial doses, 1.3 and 2.3 milligrams per kilogram on a weekly basis. Now, the most common adverse response was cytokine release. We noted that the prevalence as well as severity was on first dosing, generally manageable, and in subsequent doses, both the prevalence and severity dropped significantly. Now, one thing to note, this is a very heavily treated population, is severely ill, median 3 prior therapies, 86% refractory to their last therapy, 53% categorized as adverse risk. As noted, CRS is most common toxicity. This program is on partial clinical hold. We're working with the FDA, and we'll continue once we get this resolved. We are working through dose escalation, dose optimization for continued enrollment of this trial. This program is part of our Novartis collaboration, which was the second bispecific collaboration we entered into. Under this, Novartis received ex-U.S. commercial rights 14045. We retained U.S. commercial rights. We have up to $325 million in milestones for this program, including $90 million development milestones, low double-digit royalties on ex-U.S. sales. We retain U.S. commercial rights. We are developing this 50-50 with Novartis and sharing costs equally. In addition, under the collaboration, we're applying our bispecific technology up to 4 Novartis target pairs. They provide antibodies to us. We apply our technology, return to them. They're responsible for all development. And as I pointed out earlier, the plug-and-play approach allows us to do that type of effort with limited resources on our behalf. In addition, they also have non-exclusive license to Xencor FC Technologies, our Xtend, our cytotoxic, for up to 10 programs. Received $150 million up front and up to $2 million potential milestones. Now, in addition to the CD3 bispecific suite that we've developed, one of the benefits of the platform we generate with our bispecific is the opportunity to expand this into other areas. And we're using this to develop checkpoint co-stimulate bispecific to promote selective T-cell targeting. Now, we have 3 candidates we're taking forward, our tumor microenvironment activators, and the goal behind this is to use the bispecific to dual target more selective T-cell engagers. The first one is XmAb20717, which is currently in a phase 1 trial, and the goal there is to find, taking the 2 most validated checkpoint receptors and targeting only those that present on both by having a more selective, targeting both receptors. By having a more selective IO candidate, we believe that the potential is to have a safer version of the existing IO therapies. In addition to 2717, we have 2 additional candidates that have open INDs. One is our PD-1 ICOS, XmAb23104. This is a novel checkpoint co-stim pairing, and the thesis behind this in the animal models, this one showed the highest activity. As our CSO said, it lit up like a Christmas tree. So that's the thesis behind this, and we expect to start phase 1 trials for this in the first half of this year. The third checkpoint tumor microenvironment activator is XmAb22841. This is CTLA-4 LAG-3 that will be on top of a PD-1. So it'll be a triple checkpoint blockade. So it'll be a novel approach to taking Nivo or Pembro on top of our CTLA-1 LAG-3. And again, we have an open IND for this, and we'll start phase 1 trials first half of this year. Now, the bispecifics we've shown, we have a couple applications. We got our CD3 bispecifics, we got our tumor microenvironment activators, and most recently, we used the bispecific FC to generate a novel IL-15 cytokine candidate, and this is the basis for our Genentech collaboration. Now, IL-15 is a highly active immune signaling protein. It shares the same receptor as IL-2. The difference being is it's not bind to CD25. So what we did is we took IL-15, we built off our heterodimer Fc domain. It includes our Xtend technology for longer half-life, and we down to the potency, reduced the potency of IL-15 By down-tuning the potency, giving it a longer half-life, we have a longer duration of action of IL-15, and we believe we'll have more druggable versions for IL-15. Now this candidate, our first IL-15 candidate is XmAb24306. Under our collaboration with Genentech, we received $120 million up front. We're going to, in addition to licensing the rights to 24306, a 2-year collaboration with Genentech to identify additional IL-15 candidates. We share 45% in all development costs, but we also get 45% of net P&L on all approved products. So we retain significant economic rights in this program. In addition to the $120 million up front, we receive up to $160 million development milestones for XmAb24306 and up to $180 million development milestones for each new IL-15 that's generated from the research collaboration. The IND for XmAb24306 will be submitted by Genentech. We'll work with them, expected to be submitted before the end of the year. An important part of this collaboration is we retain the rights to perform clinical studies with the IL-15 cytokine for our own program, subject to certain restrictions. So this allows more or less kind of have our cake and eat it too, more or less. Now this slide shows our expanding bispecific pipeline. Now there's a couple of key points on here. This shows all 3 flavors of our bispecific. We've got the T-cell engagers, our CD3. There's 4 between us and our partners. Our dual checkpoint co-stimulant engagers, there's 3 of those. One's in the clinic, 2 more going. And most recently, our cytokine FC, our IL-15 candidate. In addition to the 3 different flavors, you'll notice there's multiple partners between programs advanced by Xencor, with our partners, Novartis and Genentech, as well as programs being advanced solely by our partners that built off our bispecific technology, which means we've developed a candidate, turned it over to them, and they're advancing it. And that's the 2 Amgen programs. In addition, we most recently announced a collaboration with Astellas. So we're really excited about the opportunity to continue building off this, taking these compounds moving forward, adding additional candidates to the slide, as well as additional partnerships. And this is really where we see the future of the company. Now I want to spend a few minutes talking about obexelimab, our XmAb5871 candidate, which was developed from our Fc gamma 2b platform. Obexelimab inhibits multiple passive B cells without killing B cells, or our CMO calls our non-depleting depleter. Now we've completed 2 phase 2 trials for obexelimab, one in IgG4-related disease and another one in lupus. We presented final data for the IgG-related disease at ACR in November 2017, and we presented top-line data from our lupus trial at ACR in October 2018. Now, B-cell inhibition depletion has been shown to be effective in a number of autoimmune diseases. Between B-cell depletion for rituximab, used off-label for RA, MS, and others, as well as B-cell growth inhibition of Benlysta lupus. Now, what we're trying to do with obexelimab is find a sweet spot between the safety of Benlysta, which isn't as effective as it'd like to be, and the effectiveness of rituximab, which is effective but can cause long-term immune suppression by depleting the B cells. Instead of depleting the B cells, we're inhibiting them, so we believe it can be as effective but a much safer profile. Now, these trials were conducted using IV formulation. We have subsequently completed a subcutaneous formulation and done bridging effective studies. All future trials will be done using subcutaneous injection. So we think there's a good market for a B-cell inhibitor with a subcutaneous formulation. Now, in a phase 1 healthy trial, we did an anti-tetanus challenge to show that obexelimab does have immediate and reversible decline in periphery B-cell counts. Patients were dosed with anti-tetanus, received either obexelimab or placebo, and down here you see immediate suppression of the B cell. The B cells come back, so it is reversible. You can contrast that with, say, Rituxan. Significantly depletes the B cells, and it can take 6 months or longer for the B cells to come back. So with this, we took a look at the various potential indications large or small for a drug with this patient, with this target profile. And we decided to focus on two indications, one IgG4-related disease and another one SLE lupus. IgG4-related disease is a newly defined disorder that's caused progressive organ damage, and there's no approved therapies. It's a multiple organ disease with common histopathology. Patients present on a number of different organs. Typically, it's RA or GI docs that identify it. There's no approved therapies. Standard care is high-dose steroids. We believe where this is growing is still undiagnosed. We believe that it's about 40,000 patients within the U.S. Now, for purposes of the trial, we used the IgG4 responder index to establish the potential efficacy of obexelimab. The data from the phase 2 study of IgG4-related disease show that obexelimab shows activity in this trial. Basically, 15 patients completed the trial, and what we did was we scored the patients on the responder index before the trial and then after receiving the dose. And here you see 15 patients, out of the 15 patients, 12 patients completed the trial. 14 to 15 patients achieved a decrease of greater than 5 in the IgG4 responder index. This scale over here shows the responder index before and after during the trial. So effectively, it shows that obexelimab was effective in disease activity in treating IgG4-related disease. In addition to the IgG4-related disease, obexelimab was tested in a phase 2 study with SLE lupus. Now, the premise behind this was a novel trial design that was prepared by Joan Merrill of Oklahoma Medical Research Foundation. Now, Joan's a KOL in this field, and she'd been looking for ways to show the potential efficacy of drugs, very frustrated with the number of drugs that failed treating lupus. Essentially, what this is a withdrawal trial. All the background meds are taken away from patients. They're cooled down to a steady state with steroids. And once they receive steady state, they're randomized 1-to-1 obexelimab to placebo, and they're watched over time. In an investigator-sponsored trial, Jonah observed that between 93 and 98% of patients under this scenario would flare within 6 months. So using that as a baseline, what we're trying to see is what was the effect of receiving obexelimab on time to flare as well as severity. Now, 104 patients were randomized to 20 U.S. sites, presented top-line data from this trial at ACR in October 2018. The data shows that obexelimab shows disease-modifying activity in a randomized phase 2 study. It did show positive trend in the primary endpoint proportion of patients without loss. It was not unfortunately, it did not meet statistical significance, but it did on the SLE/AI or the lupus disease activity index showed increase greater than 4 points or BILAG A or B score and physician intent to treat with rescue medication. So it did show a trend, but it did not meet the primary endpoint. It did meet the secondary endpoint, which was the time to flare or time to loss of improvement. So basically, patients on obexelimab lasted longer before flaring than patients that received placebo. So we believe this trial shows very, very strong potential for obexelimab in treating lupus. Based on these trial results, we think the best way to advance this program was with a partner to explore additional indications before moving into larger pivotal trials. Finally, I want to wrap up. This shows the milestones and goals for 2019 for the company. Primary is work with investigators and FDA resolved partial clinical hold, phase 1 for XmAb14045. That's ongoing, dialogue with the FDA. Initiate phase 1 studies for second and third tumor microenvironment activators, 22841 and 23104. We expect both of those to start clinical trials first half of this year. Finally, support Genentech's IND application for XmAb 24306, which we expect to be submitted by the end of the year. We will also have continued readouts from our bispecifics, XmAb 13676. Our CD20/CD3, we'll have data for that in the second half of this year. We'll also have preliminary data from XmAb 18087, our somatostatin receptor 2 CD3 bispecific. And finally, initial data from our first tumor microactivator 2717, or PD-1 CTLA-4 in solid tumors. We'll have preliminary data from these three in the second half of 2019. The company continues to maintain a very strong balance sheet. At the end of 2018, we had $530 million. That excludes the $120 million up front from Genentech we'll receive in the first half of this year. With over $600 million in capital, we expect to have runway beyond 2024, well into 2025. With that, I thank you very much, and I guess we have a couple minutes for questions. No questions?
Have you run across any changes in the antigen makeup of these tumor cells out of possibility?
Excuse me?
How do you choose the antibodies that you select to target a particular tumor or PA?
Oh, for our bispecifics?
Yeah.
Okay. The question was, how do we select the tumor targets that we decide to advance? I think it's a number of different ways we look at it: potential market size, opportunity, the ability for the antigen to express. We'll do a number of different animal models on a number of different candidates before we take them forward. Competitive landscape, intellectual property. There's a number the fact that we can make a lot of them means we can try out a lot, but before we even get there, we're going to look at a number of different potential candidates. Like I said, what's the landscape? What's the IP? What's the potential ability? The fact that we can make a tumor a bispecific doesn't mean it's a good target or a good candidate. So there's a lot of different factors that come into play. Any other questions? Yes, ma'am.
For that, do you also look at the reported, like the Western form and not that form included? And also my question is why this is.
I didn't hear the first part of your question, but I think the question, I can answer the second part of your question a little bit easier than the first part because I didn't hear the first part. But lupus really was, B-cell inhibition has always been, I think, rituximab off-label use in the depletion of B cells has shown that it's, the B-cell thesis is somewhat validated. And the reason we actually did it was more because of novel trial design. The fact that we were able to do a small 100-patient trial design that shows potential activity. We probably would not have gone into it using a traditional lupus trial where you do 200, 300 patients, you wander the desert for 2 years, and you come up with data that's ambiguous, right? So it's basically the B-cell thesis in lupus is somewhat validated, and the fact that we're able to design a novel trial design that's fairly effective and efficient for us to do. This was like a 2 or 3 year trial, was not too expensive to do that, and generates early but compelling data. So I didn't hear the first part of your question, but.
Oh, yeah, there's further question on like aside from systemic lupus, do you also, because you're not also treat lupus like versus for the lesser form of the systemic SLE lupus?
That one I can't address. I'd have to direct you to somebody else in another company. I'm sorry, what? Just an SLE. Any other questions? I think we're just about out of time. Thank you very much.